Let's learn together the chapter Sexual Reproduction in Flowering Plants. So the flowering plants is otherwise called as, or technically, scientifically termed as angiosperms. Keep that in your mind because we'll be saying that terms or repeating that term, angiosperms. Angiosperms are nothing but the flowering plants. Okay. So we're going to talk about the sexual reproduction. So what is meant by sexual reproduction? Sexual reproduction means it is the process of fusion of haploid gametes. So what are the haploid gametes? The male gamete and the female gamete. So these both are haploid. Haploid means they have only one set of chromosomes, that is n in number, one set of chromosomes. We represent the number of chromosomes as n, or the set of chromosomes as n. So gamete with n number of chromosomes fuses with another gamete with again n number of chromosomes, and this both fuses, so n plus n, it is a diploid one. So it results in the formation of a diploid. Diploid means 2n. So it has two sets of chromosomes. Diploid structure or cell, which is termed as zygote. And this zygote develops into embryo and it further forms the new organism. So this kind of reproduction where two haploid gametes fuses to form a diploid one, we call it as a sexual reproduction. So we are going to talk about the sexual reproduction in flowering plants, that is angiosperms. So in flowering plants, which is the reproductive part, or which is the reproductive organ, or where the reproduction takes place or occur? Yes, it is flower. So in flowering plants, flower is the reproductive site or reproductive organ. So first we have to learn about the structure or parts of a flower. Let's learn the parts of a flower. This is the structure of flower shown in your textbook. So let's zoom it and look at the structure. So this part, the basal part, we'll start from the basal part. So we used to say that a flower has 4 whorls. A typical flower has 4 whorls. We call it as whorls. The lowermost or the outermost whorl is made of sepals. And we call it as calyx. The second whorl is made of petals and are termed as corolla. C-O-R-O-L-L-A, corolla. The next whorl is made of stamens, which is otherwise called as, or together called as, androecium. A-N-D-R-O-E-C-I-U-M, androecium. And the innermost whorl is made of pistil, or sometimes they use the term carpels. And together, all the carpels together, we call it as gynoecium. Okay, so these are the parts of a flower, or 4 whorls of a flower: calyx, corolla, androecium and gynoecium. Calyx are made of sepals, corolla are made of petals, androecium consists of stamens, and gynoecium made of pistils or carpels. Now, you can see, we'll see this diagram. Hope you can see the diagram clearly. You can see the sepal, this whorl. Okay, so there will be many sepals together. We call it as a calyx. That whole whorl we call as calyx, and the whole whorl of petals we call as corolla. And this stamen, this part, these parts are stamens, which is mentioned as anther and filament. Together we call as androecium. And the last one, this is stigma, style and ovary. Together we call as pistil or carpels. Now you can see all these structures are arranged on a base or a receptacle, and that receptacle portion is called as thalamus. There is one more term mentioned here, thalamus. Thalamus is the receptacle or the structure or on which all these 4 whorls are arranged. Now, if a flower has all the 4 whorls, we call it as a complete flower, and if it don't have all the floral whorls, we call it as incomplete flower. So the one with all 4 whorls, we call it as a complete flower, and the one which do not have it, if it miss any of the whorls, we call it as incomplete flower. So you may wonder, how can these flowers miss any of these whorls? See, sometimes some of the flowers will be having either stamen or pistil. Either stamen or pistil. In that case, we call them as unisexual, because stamen is the male reproductive part of a flower or male part of a flower, and pistil is the female part of a flower. So if the flower possesses either male or female part either, then we call it as a unisexual flower. And if a flower has both the parts, then we call it as a bisexual flower. So this is what you have learned in class 10 too. Unisexual and bisexual flowers. So you can just now you can imagine that a flower which is having either stamen or pistil, so one of the whorl is missing, then that kind of flowers are incomplete flowers. Okay, so unisexual and bisexual flowers. Is it clear? Now let us move on. Details of the parts of the flower. We are not talking about the sepals and petals. The sepals and petals, they play a role. They do play a role in reproduction, but not directly. They do enhance the process of reproduction, like sepals which are green in color, which prepare food and protects the flower in the bud condition, whereas the petals which are colored and with fragrance may attract the insects for pollination. So such functions they do serve. The major or the organs or the parts which directly take part in reproduction are the stamens and the pistil. So we're going to talk about the stamens and pistil in detail. This is the figure of stamen given in your textbook. You can see that they have taken one stamen out of this. There are many stamens, and from this we have taken one stamen out. So stamen, this is the male reproductive part. So we are going to talk about stamen, which is the male reproductive part of a flower. Now you can see stamen has 2 regions, anther and a filament, which is a stalk. So it has 2 regions, anther and a filament. Filament is the stalk with which it gets attached to the receptor, and this anther is a bilobed structure. You can see it has 2 lobes. So it is the terminal bilobed structure, which is mentioned as terminal bilobed, which is at the top. Okay, and this one, terminal means it means the end. And here this region is considered as the proximal end. Proximal end is the end which is getting attached to the thalamus. So the proximal end which is attached to the thalamus is the filament. Okay. So filament is the proximal filament. At its proximal end, it is attached to the thalamus. So I emphasize this because few years back they have asked this question as in multiple choice, and one time they were asked as assertion reasoning. So filament at its proximal end is attached. Proximal end, this one, this is the proximal end here. In this case, this is the proximal end. Okay, with the help of proximal end, it is getting attached to the thalamus. So you can see that thalamus is here, and this thalamus filament is attached to the thalamus at its proximal end. It is mentioned as it is a bilobed structure. You can see here it is a bilobed structure. It has 2 lobes, one lobe here and the other lobe here. You can see the 2 lobes here. Fine. Each lobe is having 2 theca. So this is a lobe. This is one lobe and this is another lobe. Now each lobe is having 2 theca. Theca means chamber. So this is one theca and the lower one is the other one. So one lobe has 2 theca, and again the next lobe has 2 theca, right? And so this lobe is actually dithecous. Each lobe is dithecous. You understand the term dithecous? Because it has 2 theca. We are talking about each lobe. Okay. Now, if you take the cross section, now we will be wondering that it has 4 structures. So we can say stamen or the anther on its cross section, they are tetragonal. Tetragonal means it has 4 structures. Now, inside this theca is a structure called microsporangia, and this microsporangia will later develop to pollen sac or will become pollen sac, and the pollen sac carries the pollen grains. That's how the development is going to happen. Okay, pollen grain. So the male structure which holds the pollen grains are called as microsporangia. Microsporangia, which is having microspores or which produces microspores. Clear? That microspores develop into gametophyte, and then gametophyte produces gametes. That's how the development occurs. Sporangia produces spores. Spores develop into gametes. Gametes give rise to, or gametes contains sorry, and that's how the development occurs. Sporangia produces spores. Spores develop into gametophyte. Gametophyte carries gametes. Clear? Now let us see in detail the structure of microsporangia. So you can see this towards the, I'll just zoom it out. You can see this region are called as the lines of dehiscence. You can see the pollen grains comes out through this region. So this region is called as the line of dehiscence. The pollen grains are coming out. You can see that. Okay, the pollen grains will be coming out. So this is one theca, another theca, so dithecous, bilobed, and since it has 4 structure, we can call it as tetragonal. On its cross section, we can see some 4 structures, so we call it as tetragonal. And what is this actually? There are 4 microsporangia. Now let's study the structure of a microsporangia. So we came to know that in one anther there are 4 microsporangia. Okay, microsporangia here 1, 2, 3 and 4. So let's see the structure of a single microsporangia. Now in this figure you can clearly see 4 microsporangia here, that is tetragonal structure of an anther. Now here they have magnified this part of one microsporangia. So when this part is magnified, you can see there are 4 layers on the microsporangia, 4 outer layers. Okay, so each microsporangia has 4 wall layers. So what is the first wall named? Epidermis. Second wall layer is named as endothecium. Single epidermis, single endothecium. Then we can see few middle layers, and then inside or next to middle layer we could see the special layer called tapetum. So these are the 4 wall layers of microsporangium. These first 3 wall layers are protective in function. Their function is to protect, and the innermost layer, that is the tapetum, it helps in nourishing the developing gametophyte. It helps in nourishment. It nourishes. Okay, the outer 3 layers, epidermis, endothecium and the middle layers, are protective in function. Also they helps in the dehiscence of anther. The anther must dehisce, right? The anther should break open to release the pollen grain. So it also helps in the dehiscence of anther. Dehiscence means it breaks open to release the pollen grain. So these are the functions of the outer wall layers. Now if you look at the structure of tapetum, it has nucleus and dense cytoplasm. There is a possible question in your textbook and this chapter. Can you think of how tapetal cells could become binucleate? So we have mentioned that tapetal cells can be of binucleate. So the chance of tapetal cells getting binucleate is that karyokinesis, when the tapetal cells are formed, the karyokinesis process may not be followed by the cytokinesis. Do you have any idea about karyokinesis and cytokinesis? During the cell division, the division of nucleus is called as the karyokinesis, and the division of cytoplasm is called cytokinesis. So while the formation of tapetum, after the nuclear division, sometimes the cytoplasm may not divide. Cytoplasm fails to divide. In that cases, there is a chance of tapetal cells getting 2 nuclei in it, and that's how they become binucleate. Inner to this wall layers, there are cells here. So we have focused or magnified or highlighted only this region. So inner to this, they have the sporogenous tissue. So these sporogenous tissue will give rise to the pollen grain. So let's see how the pollen grains are formed from the sporogenous tissue. Now you can see a structure called connective here. This connective is nothing but some kind of sterile tissue. Sterile means they do not develop into any of the reproductive structures. They are a kind of normal tissue, sterile tissue, which act as the connective between or structure between two theca. And probably if this anther has got a filament, the filament is attached at this region. Okay, it is the region at which the filament is attached. Now let us see the development of pollen grains from the sporogenous tissue. So the sporogenous tissue are the one which is found inner to the wall layers in a microsporangia. The sporogenous tissue, they are diploid. Okay, the sporogenous tissue are diploid with 2 sets of chromosomes. This tissue will undergo a process called meiosis, which is the reduction division. They undergo meiotic division, and as a result of meiotic division, it forms 4 microspores. As a result of meiotic division, 4 cells will be formed, 4 microspore tetrad. These microspore tetrads are otherwise called as microspore mother cell. Also, the sporogenous tissue, each cell of the sporogenous tissue is otherwise called pollen mother cell, which is abbreviated as PMC. These microspore tetrads will give rise to microspores which develop into the pollens, which further develop into pollens. So these pollens are the male gametophytes. They are called as the male gametophytes because inside this male gametophyte they carry the male gametes. And the development of this development process, that is the formation of microspores from sporogenous tissue, is called as microsporogenesis. You got it? Sporogenous tissue will undergo meiosis and form 4 microspore tetrads. Microspore tetrads give rise to microspores, and microspores further develop to form pollen. And we know that this is haploid. Microspore tetrads are haploid in their number, chromosomal number. So this is microspore tetrad. 4 cells you could see here. Once this microspore tetrad mature, or once when the anther mature, these microspore tetrads dehydrates. They lose water, they dehydrates, and they dissociates. Dissociates means they get separated. They dehydrates, they dissociates, and they separate to form 4 viable microspores. They are in tetrad form, right? So all 4, each one are viable from each tetrad, and each microspore develop to form the pollen grain. They mature or develop to form the pollen grain. That's what you see in this picture. And once they develop into pollen grain, they will possess the male gamete. So this is called the gametophyte stage because they possess or they carry the gametes or the male gamete. Okay, this structure is actually pollen grain. Now let us carefully observe the structure of pollen grain. To learn about the structure of pollen grain, let us enlarge this picture. If you look at the pollen grains of different plants, of different flowers under the microscope, it will be really beautiful to see the pollen grains of different colors, different shapes and all. Basically, the structure of pollen grain has 2 layers. You can see 2 layers here. Okay, the outer layer is called as the exine, and the inner layer is called as the intine. The outer layer, exine, will be having a lot of spinous structures. It varies from species to species. So that spinous structure will help the pollen grain to stick onto the stigma. And this exine layer is actually not continuous. Exine layers will be discontinuous. Okay, that discontinuous part of that exine layer is actually called as germ pore, and it facilitates the germination of pollen tube. Do you remember in class 10 you have learned during fertilization pollen grains germinate to form a pollen tube? Yes, that pollen tube comes out through this germ pore. So we were talking about the layer exine, right? This exine layer is made up of a special kind of organic material identified as one of the most resistant organic material, and that material is called as sporopollenin. So it is identified as one of the most resistant organic material, and so far no enzymes have been identified which could dissolve this sporopollenin. So now I think you can realize like how strong they are, okay, how resistant they are. And that's why this pollen grains could be stored for many years. Even in the fossils, we could find the pollen grains remains active, or it can be stored, okay, in the fossilized form. And even it can even withstand high temperature, strong acids. All these will not affect this sporopollenin or affect this pollen grain due to the presence of sporopollenin. Okay. And as I said, so imagine this is a sporopollenin. They are not continuous, so there'll be gap in between, which is called as the germ pore. Inner to the exine is the second layer called intine. Intine is actually thin and continuous layer. So exine is actually discontinuous layer, and intine is thin. This is the second layer, intine. It is thin and continuous layer. Okay, and inner to the intine is the contents of the pollen grain. So it has a cell which has a plasma membrane. Inner to the plasma membrane there is cytoplasm, and inner to the cytoplasm there is a nucleus. And this nucleus will divide mitotically. Mitotically means there is no change in their chromosome number. It's already haploid. So they will not further undergo any meiotic division, but they undergo mitotic division to form two separate cells. They form two separate cells. One is generative cell and the other one is vegetative cell. Okay, so let's see in the diagram how do they divide. So in this diagram they show that it is a typical cell with dense nucleus, cytoplasm, vacuoles, and now this nucleus is undergoing mitotic division by the formation of spindle fibers, and this spindle fibers is asymmetric. Why the spindle fibers, and what does it lead to? It lead to the formation of two different kinds of nucleus. Then usually when a cell divides, the nucleus will be separated by symmetrical spindle fibers, so that the nucleus separated will be of the same structure. But in this case it is of asymmetric spindle, and you can see they formed two different cells. In one cell, the upper one, which you see is the bigger one, is called as the vegetative cell, and the lower one is called as the generative cell. This vegetative cell is usually bigger and it has huge food reserve. Quite a lot of food reserve is there, and the size of the nucleus is also bigger, irregular nucleus. Whereas the generative cell, this would be spindle shape, and it will be found to be mostly floating in the cytoplasm of the vegetative cell. Actually, the gametes are formed from this generative cell. This generative cell will further divide to form two male gametes. So this generative cell is actually dividing to form two male gametes. See, usually in most of the angiosperms, that is, it is mentioned that almost 60% of the angiosperms, pollen grain is released out at this two cell stage with one vegetative cell or two nucleated stage. That is a nucleus of a vegetative cell and a nucleus of a generative cell. But for 40% of this angiosperms, the generative cell, the nucleus in the generative cell further divide, pollen grain is released at the three nucleated stage. So imagine this is a nucleus of the vegetative cell, and this generative nucleus will divide into, so mostly in 40% of the angiosperm, it is released at this three nucleated stage. Okay, three nucleated or three cell stage. These two are the male gametes. At this stage, the pollen grain, or at this stage, the pollen grain is released out of the sporangia. It is dehiscence of anther releasing the pollen grain. Now let's talk certain significance of pollen grains. See, pollen grains may have some harmful effects, like if a lot of pollen grains are released at a time, it can cause allergies, like some bronchial infections, respiratory disorders like asthma, bronchitis, etc. So if you just see about the harmfulness of pollen grains, it's not always harmful. certain cases, certain plants, some plants like Parthenium, if the pollen grains are released together, it may cause allergy. It may cause allergy and lead to respiratory disorders. Respiratory disorders like asthma, bronchitis, etc. This Parthenium is not a native of our country. It is supposed to be, this Parthenium has got imported along with the wheat. And now we find it very difficult to eradicate this weed. Now, if you just see the usefulness of or the significance of pollen grain, this pollen grain is actually very much rich in nutrients. Okay, they are rich in nutrients, and you can see in this figure, you can see that bee pollen, it has come in tablets. Okay, so we can use it as food supplements. It improves the performance of, it's found that it improves the performance of athletes and sometimes even the race horses. It improves their performances. And we know that we have discussed in the previous slide that sporopollenin found on the egg sign will help them to remain undisturbed or undestroyed for many years, right? So this pollen grain can be stored in pollen banks and all for many years and can be used for crossbreeding programs. It can be used for crossbreeding programs. So what helps them to get stored? Actually, they are stored cryogenic storage. That is, they are stored in liquid nitrogen at minus 190 degrees Celsius. They are stored in liquid nitrogen, and then so they can be stored for years together because the sporopollenin keeps them active. And this pollen grains must be used when they are viable. Viable means when they are active. So this viability of pollen grains varies. In some cases they will be viable for many years. In some cases they will be viable only for few minutes or hours. Okay.